Let's get started. Hi, everybody. It's my pleasure to introduce Manyuan. He's a distinguished professor at the Ecology and Evolutionary Biology Department of the University of Chicago. Uh, Focused on studying the evolution of new genes in fruit flies, but also in other systems as well, which is a really exciting topic. And here today, I think we'll hear about the evolution of new genes as they affect males and females. Dr. Long has received a prestigious Guggenheim Fellowship and is a AAAS fellow as well. And I'm really excited to have him here today for our genetics colloquium.
Thank you, Nathaniel. Thank you for Nathaniel for invitation. I came to this place the other day, seeing my schedule, which lists more than many people's office address in genetic. Remind me, I come from serious genetic place in the country. So thank you for your coming for my talk. So today I'm going to briefly talk about introduction before I discuss our recent work. Some of them unpublished, someone going to publish and share with you to discuss. And first, I would like to present in what kind of historical context we talk about the topic of new gene evolution. That's very interesting. And these two are probably very famous, and they founded molecular genetics, molecular biology, and in a way, they were a leader of a scientific revolution in last century. They give a lot of great contribution. They also comment about the new gene evolution. Basically, they're very negative. And like Francis Jacob, he liked to say the probability of functional protein would appear de novo by random association amino acid is practically zero. It's impossible. And understand the French scientists impact by the famous, philosophy of Cartesian philosophy, the world is a machine-like world, so impossible to change the new gene, the impact by that. And the person who have most direct impact, big impact to the field is Alan Wilson. He was the person who proposed out of Africa hypothesis and in the genetic, and unfortunately he left earlier. So in his writing, he claimed that gene duplication is not essential. Only regulated element is evolved. I know this idea deeply impact the later great scientists like Shankara used to be here, the impact that kind of idea. He was the first one propose the role of regulated element to evolution. They don't believe that the new protein, new gene can still today generate it. And the person who directly connects my work is the former Cambridge Department Chair, Michael Ashburner. He made very straight claim. He said essential genes are not species specific. In other words, if a gene are important, that got to be very old, got to be very ancient. And Meyer, of course, evolution biologists worship him as a grandpa of biological species concept. He picked up the idea, Michael Ashburner, Alan Wilson, and also Wilson stayed the same in his famous book. Just said that it's impossible. Even new gene duplicate evolve new function. So the textbook, two most famous one, I give one example. One is edited by Jim Watson, Molecular Biology of the Gene. Here, everybody read it in certain time. And also Benjamin Lewis' gene right now, 14 or 12, something like that. They both summarize all this pioneer idea that is new function or new important function, impossible to create it today. All this historical background, we are working at Chicago and with some colleagues right now in the country. And we showed that all this idea, unfortunately, was wrong, or fortunately, for our own pursuit of the science. I give brief overview for most several important steps before I present our recent observation. First one is an overview and the evolved new gene and the function. I wrote some review recently. Of course, first work we call the gene. which was a mistake made by Michael Burr, and then we corrected, show that that is really the first new gene in 1993. After that, we are in different times. So today, we're in a genomic era time. Here is you summarize 35,433 species genome. You mapping about that. Immediately you find that. You talk about dimension of genome size, then there is almost more than 9 order magnitude difference among the life, different species. You look at the gene number, they also differ by 5 order of different there. So today, when you're facing this kind of distribution, this kind of observation, you have to believe, you have to say, compared to evolution tree created by Charles Darwin, that is origin of new gene is a general phenomena, is one of the most general evolution process. We can draw that conclusion right now. Especially for us, like here, somewhere there, you see that our ancestors step by step pick more and more new gene to make you and me sitting here. So from there, we can define what's called a new gene mean that. Those genes just appear from some locus. Previously have no genes, suddenly some new locus appear in some technology. And what kind of method? We usually teach students that from Hermann Mahler's work, gene duplication either way to create a new copy, which might evolve some kind of new function. Thanks to the effort of entire molecular biology in recent several tens of years, today we identify at least 14 different kind of mechanisms. That is, there is more than a dozen mechanisms more scientists didn't know before, only know gene duplication. So today I'm going to show you about 4 mechanisms. One is retrocalation, other one is gene duplication, other one is de novo origination. All other mechanisms right now have degree of understanding, but most of them we don't know much. We just know the mechanism is there, so provide great opportunity for students or people in the future to understand what molecular mechanism, what genetic mutation can create the new gene. I'll give one example. So several years ago, we reported in the grass, in rice, there are more than 350 new protein which come from de novo, from non-coding region from 500,000 years ago to 1 million years ago. Everything is so strong for each of 350 de novo gene, you can reconstruct their evolution step, show that how the stepwise gradually the gene created most recently from non-coding intergenetic region today have a brand new protein. So this was really changed our view about how protein structure evolved, how gene was created. They created before. Give one example. So like this new gene, we called in the chromosome 5, gene 30030. You look at align the gene together, you see that in the first place, when you see outside of a group in this species, which diverged from about 1 million years or 2 million years ago, it was stop codon. You would assume that until stop codon gradually was replaced in this movement, in the event 1, when a substitution happened to make TAG into CAG. So in that way, the breakthrough create a new protein longer until stop here. In the second event. In the second event, all of the codon have 2 nucleotide deletion. That deletion could make a gene create long code or create just stop. Long code just stop here, no longer. But then some nucleotide insertion happen to move uniform again, step by step. Today it becomes standard protein coding gene there. And so that shows that what kind of evolution event happening during the process, which can turn some long coding RNA, then become a shifted new protein. And in the genomic era, we created computational biology using parsimony principle by looking at the synthetic structure in the micro level and macro level to determining the homologous stature of the gene, like the gene 1, the 2 in your orthogonal position. You do global alignment, then you scan, you write out this is new gene in the middle, which is origin after species B diverged, before species C and D diverged to create a new gene there. So in this way, you can quickly automatically reading the age of every gene in a genome. So this is really great time to do evaluation. Now I just mentioned the gene, which is first new gene was actually identified from this group. At that time, there was no genomic sequences, but the DNA fragment analysis at that time, 1993, allow us to identify the first new gene, which evolved a biochemical function and to synthesize new hormone and a new metabolite hormone to help Drosophila to communicate in different ecological environments. And so right now, this is not secret. And almost every species you can write down the age of every gene, as long as you have the genome from most related species, then you can identify those called the new gene, which might be 10 million years old, maybe a few million years old. Here, the picture simply shows all major organisms from human being to mouse to Arabidopsis to Drosophila to nematode to fungi. You find that in the recent evolution time, they pick up a new gene quickly and they increase biological functional diversity. So that is right now. So that's why I say this is a common phenomenon, one of the most common phenomena for the gene origination. I also thank the effort of great molecular working all this half century. They work out the gene function for many genes. Then when we look at the literature, you find that a lot of well-defined function is actually the new function of recent evolved gene there. So those functions, I read the list in the review, including all kinds of functions, mostly about neurological brain and reproductive biology, adaptive, all kinds of function there. So we check about more than 2,000 literature, cite about 400 people there as an example. So today that we not only prove neurodegeneration is a general phenomenon, we also prove that many of them could have important essential function. So that is what today called the functional diversity, which is support the great diversity of phenotype in the live organism actually is provided by the new gene and the recent evolution history. They're not the only function, they also have special pattern. One of the patterns we like to call is out of testis rule. We can call it rule or hypothesis. So there we map some age of the gene in Drosophila in different evolution time from the recent 30 million year or 0 million or today or 44 million year, mapping their time. Meanwhile, we were able to calculate their expression broadness. What do you see that when the gene is very young, if the gene is like a few million years old, then you know that they must just acquire testis-specific expression pattern. When the evolution time becomes older and older, you're going to see that their expression breadth across tissues more and more. And when the gene is very old, like more than 126 million years old, then they basically express in many tissue from male and female. So as we showed there. So i.e., expression pattern for new gene, most of them originated from this bar, which we biologically call the testis. They always go from there. From plant, like from rice to other organisms, they usually started from anther. So that You have to make connection between the plant male organ and the animal male organ in terms of how gene originated. And so today, and most often we want to focus how the gene contributed to the fitness to get species evolution, that one of the most important phenotypic trait called the fitness effect there. So first I'm going to present one case we published some years ago. Then I'm going to present what we found recently at the genomic level and drawing a big picture. So before our work, including ourselves, we try to guess if we tested the fitness effect from what kind of fitness is going to be collected. We might guess that the new gene function or functional innovation must be designed, if we are able to co-design to solve adaptation problem, to help organisms to evolve, to adapt to changing environment. That is the conventional idea in most people's mind, full of all kinds of publication and newspaper and stories. For example, like antifreeze protein is a good example that's really helped species in recent 3 to 80 million years. They evolved to help species to survive in the colder, cold sea water. Is that true? Is that general? Our study showing that no, that's not the case. So first detailed case study done by my student Nick Van Curen. At that time, he's interested for single gene duplication, say tandem duplication, how they evolve quickly into new function, which we call Artemis and Apollo. What is evolution force? What is evolution force? I'm going to drive the gene movement by looking at the gene knocking out function. So the gene was identified as a tandem duplicate from 1 copy into 2 copy. And today, when you compare the 2 gene sequences, you identify that they change protein sequence frequently in this region, but they more frequently change the frequency in the 5' reader, promoter region and enhancer region, and also non-coding and intron there in the region there. So that was sequence analysis. You might see that because this gene is only contained in the genome one single species, Drosophila melligetter. You might think there might be some kind of polymorphism, nothing more than that. Then using 97 line John Poo isolated in Africa, that represented basically most of species distribution. We found that every individual line have the same gene duplicate intact, suggests that the gene had been duplicated something like 200,000 years ago, which is not mutant, which is a fake standard gene there. So then at that time, we used a gene editing technology in the lab and created 100 base pair deletion in 5' region in Arboro strain. We also created another 200 sequence deletion in the Artemis in other one. And then we also isolated their control line from the same procedure from same species to make it real. We might call it isogenic background there because they're from same stuff we created, so efficient control. So then we measure its viability. Basically, you throw 50 strain in a vial to observe how many of them grow to the add up. So there we show that in such a young gene, they do have fitness component in the relative survival rate in Arboro and Artemis. And Arboro actually have more, strain cannot survive which can to about 40% to 30%. Others about about 20% or higher cannot pass the life stages. Most surprising observation, which changed our view, is like that. So we started to measure its fertility. Basically, use homozygote gene deletion to test with goes line to measure fertility rate there. First thing for the arboro, we find that where control is, say you take control as 1.01%, then the two low line lost all the fertility, such that the gene evolve essential function. And then we try to rescue it. Only recover it to the degree. So that means in the short as 20,000 years, arboro become diverge essential function they require for male. But you go further, look at the behavior, it's more surprising. So in this gene, when the gene is absent, female actually have significantly higher fertility rate compared to the control line. So I was really shocked. I said, just do this thing again, I'll repeat again and again. Then when you rescue Drosophila provides the most serious genetic, you can rescue it, put those wild type of gene into this low line to say can they recover their function there. So they can recover. They can recover to about 1%, or close to 90% to 1%, about there. So that is first time they show that means those female actually suffer in nature in terms of its potential for reproduction. It's the first time that I was really shocked, my student. That means what we call the wild type have some gene which have bad effect, but they always have a standard sign over and over again. It is significant, and by somebody doing 50 control. And so the rate is really high. The higher than you really higher than about 20% of kind of fertility, higher fitness is really high selection coefficient. But that's happened in nature. So that means arboro is under sexual antagonism selection, favor male, but hurting female. And so that is really amazing. But alternatively, you look at Artemis. When the gene is deleted, the female loses all the fertility. You see, that's there. So that means in the female, in a short time of 20,000 years, they evolve from redundant function to diverge essential function required for female. But what we unexpected is that the same gene deleted, but in the male, they are going to increase significantly higher fertility by more than 20%. That's really high. 20% to close 20%, well, that's the highest one have that one. So that means in that way, nature law is fair. While female suffer, male also suffer from to complete other part of female function. So that it means Artemis is under sexual antagonist selection, favoring female but hurting female. There you can see the effect significantly. Use the most rigorous genetic control, most rigorous rescue experiment. So put them together, it's having to form kind of symmetry distribution. But you still see that for arboro have even stronger effect, which have male advantage, male essential at cost of female fetus or female detrimental. Slightly higher than this one. In the female, it is male going to be, or it is female essential, but at cost of male effect. So what does the gene do? for the single species Drosophila melanogaster, of course, we're looking at their morphology of sperm, of the mutant, and the control. In the control, you'll see that the sperm stop development in the late individualization stage. They do not grow the tail. And normal one, just like this one, you can see that the long tail normal the stop. So such as the gene actually evolved to add a new function to let Drosophila melanogaster, today's Drosophila melanogaster, to complete their spermatogenesis. Without that, you cannot complete the species of the tree. There's complete new gene there. What about female, which is also very obvious? In female, the null line, control line, the control have very lovely, this egg and this one, but when the gene are locking down, the shape is changed. Under a microscope, you will see that the new gene actually there play a structural role, could form kind of actin layer to protect the entire sperm there. For using single cell analysis and also the RNA expression, we saw that the two genes evolve complementary expression pattern. For the arbolo, most often, likely about 100%, they evolve in the testis, which is same level of wild type single gene copy. For the female, then they most often express in the ovary. And so this one is other data just showing that same kind of expression there. And there, what we see that when you see the atom expressed in the male testis, the later stage going to lose those bad cells that are supposed to be sexual antagonist cells. Suggest that during the development, there's some way to try to resolve the conflict the gene created, but not just to mitigate it, the conflict will not really resolve. Be not resolved, we cannot see it. And so this is a model how gene duplicate evolve and from the way to eventually mitigate it resolution by gene duplication. So there are a lot of models about that. So in the same gene family, we identify 1, 2, 3, which called retroposition, showing same kind of gene pattern between male and female. So we propose hypothesis that there might be also evolved sort kind of sexual conflict with resolution mechanism. So that is something we are doing. I just mentioned that. You compare the sequence between two genes, which is just 200,000 years, but their sequence diverge as high as 2 to 3%. Given its age, this is a high frequency rate, which is 15 times more quickly than genomic average compared to David Began, Chaglani data, that 23 times more quickly than the genome average in regulatory region. So that's what tell you a secret in nature. They evolve more in regular element, try to make it more diverge, more bias, such that to create less trouble for other sex. But it's still there. The trouble is still there. And then using population genetic model, we show there's strong selection surrounding the gene region. So today in nature, this gene region is pretty homogeneous. So the age dating is about 50,000 years ago. The population, they push for gene duplicate to change sequences to resolve probably to mitigate their conflict bad effect from harmful sex. And the model we came out, we believe that this is very interesting. So first time when you got a gene, for some reason, the machine called sexual selection, going to push some male bias gene from male advantageous gene, so male positive. Then you make female suffer, called sexual conflict. But then, this is the hypothesis I propose. Then the fetus landscape in the female would change. Those mutations either is neutral or bad. Today, they might help female to survive better, to against that female effect, the male gene function to bring to. But for the same reason, when female after selection, post selection, as we tested, become positive, then they're going to make male suffer. So in this way, pretty much like MC ether picture. So there's circular picture over and over again. Then you'll see how the gene sequence evolve over to fake eventually bring the larval function. And so this is the first time we show that we're able to see that nearly experimentally, see how those sexual conflicts appear, how imperfect the world of life really is. First time remind me when I teach textbook biology, I will not tell my students this world is beautiful. We have gene A because they want to help gene B. That's not really the case. This is strong evidence showing that that cannot be the case. Right now we have to answer question: how general is the sexual conflict, what I call it sexual conflict drive on gene evolution? So we started work right now. We have done the project for 10 years right now. It's in a stage to consider to publish, to accept it. I will discuss with the data with you today how general phenomena is. So based on what we learned from Apollo Artemis, what we did is try to, well, first of all, pick about, say, 200 new genes in Drosophila. We already identified and published many times. Then we want to use the gene locking down technology to locking down expression of the gene in high density of repeat. I know a lot of people have problem with the gene locking down, high repeat, and set a multiple layer of the control. What we use technology, this RNA locking down. Everybody here in Drosophila familiar with how to do that. Basically, RNA was created for certain kind of specific gene, then it can kill the RNA and reduce the number. We show that in the Drosophila, today's locking down technology, on average, can locking down gene expression by 30 to 40% on average strain. You order from Vienna, from Harvard. So that we analyzed 70 strain there, so prepare technology so we can use it. So we set 3 layers of control, including, of course, the original strain. We use it to do those gene locking down. Then we also measure the control, the RNA line itself. Then we measure the 3 driver, including somatic driver, actin 5C, and tubulin. And not the germline driver, I want to spend more word. People usually call actin tubulin as constitutive driver. That's not very accurate word. That is constitutive driver for somatic cell, not really for germline cell. A lot of papers showing that they do not really locking down germline. So they form complementary locking down driver. So we set 3 control. So the whole journey that we choose in Drosophila lineage from ancient time to today's Madagascar, in a time less than 33 million years stage, in Sophophora subgenus, we choose about 92 genes there. Their age, some is very young, 23, some is slight, intermediate age. We also choose 33 old gene. They can be 6 million year, some of them even can be 100 million year. We call the Asian gene there. So the entire experiment is that we basically try to lock it down. In this data we presented, 125 genes in male and female, using 3 drivers, and repeat by 10 to 30 biological repeats. Some we repeat more than 40 times, independent locking down there. And then total, we did about 15,000 locking down experiments. And this is 3 major group in the lab, major team, we complete experiment. So we reported the data of 732,710 viable adult offspring. We score each one and looking at how many die, how many survive. And we also created probably another 500,000 flight data from other 70 genes, something like that. That is de novo gene, and we are still analyzing that data. So often the data, this is kind of complex control repeat experiment. So that you cannot simply use a simple calculator and to compute the result there. So we using Bayesian linear regression model, try to model that different effect, locking down effect, control 1 and 3 effect as a linear regression of the average of GNI, the average fatality effect. Then in this type, all the colleagues here to Drosophila, you know that Drosophila, when you grow the fly, you have certain kind of probability that bacteria are going to contaminate it. We have about 3% in the whole experiment. Even you control that one, you would see that the distribution of fertility actually is quite variable, more variable than Poisson process or Poisson distribution can able to distribute it. So therefore, we use a negative binomial distribution to control it. Negative contribution is kind of derived geometric contribution by counting multiple successful rate first time. So we use this model, and then to run Markov chain multi-call simulation, and use this in the R language, use a no U-turn sampler. We do four chain and repeat 3,000 times. And if one chain become converge, we use converged one. So that practice of mathematics statistics come up with 323 locking down in 2 sacks, which is total the data of 646 fatality effect for 2 sacks in 3 driver from 125 gene there. So I wish I give enough adequate details, how we doing there. And first time, what we saw that almost all the genes have locking down effect, which is as fatality effect as high, which is higher or equal to 1%. In evolution biology, 1% is really high selection because they show that 200 generation, your mutant going to become fixed. So that is, everybody agree that is one. We also see that there are about 10 locking down have fatality effect is 0.0001 to 0.0001. That I don't know I should believe theory or believe my own observation. And because in observation, we grow at the most 1,000 fly. There's no way you can really see that like 0.0001 effect because you don't grow about 10,000 fly, you will not see it. But the theory gives us that. So I was matter of fact to describe it, and my own thing, I believe my experiment. I don't believe theory in that experiment. Fortunately, that only take 1.5% of the data. So that's about the data side. And so first one, we try to map those fatalities in male, which is blue line. And there's a fatality of the female of line T young gene, which locking down by action 5C GAF4 is similar, which is somatic locking down. And because we try to compare to null hypothesis, I mean, when gene have no effect, what do you expect to say, like to follow 0-1 normal distribution? So we also plot that dashed line is a null hypothesis distribution. So for that reason, we divided the fatality effect by its average sigma, average standard error in terms of Bayesian distribution there. what do you see there is that for male, its average effect is minus 0.1. That means that is 0.1% original fitness. That's really high. They move to that one. To the male, you see the average fitness is 1.30. Let's normalize it. And the original fitness is something like 0.3%. So that means that both elements push to the two ends. That definitely is not null hypothesis can explain. And then using kind of Kolmogorov-Smirnov test, then we show that in the male, the blue line comes from dashed line, that's P equal to 0.0012. And for the female, that probability is 1.05 to 10 to minus 23 order of magnitude. Basically, it's zero, actually practically zero. Impossible that observe come from this data. So in that way, you have to believe everything is significant. When we measure the overall database, all the genes in the 646 locking down, then you see that probability for the male, that blue line comes from black line, is 5.48 times 10 to minus 9. That's big enough, or that's small enough. For the female, the probability is random, come from random hypothesis, or from null hypothesis, that's 9.01 times 10 to minus 30 order magnitude. So you would feel confident that this gene really have functional, very significant. So the hard effort we made get paid off. And so this is really high there. But from here, when you see the overlapping between the two, you see the separate the two distribution, you have to believe that there got to be a lot of gene in a conflict, and which is observable, detectable significant between this one and blue line and red line. But how small it is? So then we map those data in two-dimensional figure. One is female fetus, other one is a male fetus. And there we label those probability, red color is anything gene have selection coefficient more than 0.25. That's very high. And the green line is more than from 0.01 to 0.25 in between. That's also strong. And there's a very few data, about 10 data points. Here is about three data points, a few data points that is from 0.01 to 0.0002 in this set of data. Basically, almost all of them are significant and in the distribution. Then you start to count. In the first quadrant, which is showing male plus female plus, means both sexes doing badly in wild type. In quadrant 2, which is male plus female negative, that means the gene doing good in female, doing bad in the male. In the third quadrant, then both genes do good job, in male and female. The gene in both sexes doing good job, male and female. In the fourth one, which describes sexual antagonism, which is male advantageous and female detrimental. You count the number, that is, there's about 50% in Q4, which take almost 60%. They are sexual antagonists. In next one, Q1, this one both doing bad in both sexes, that takes 31%. How come so many genes doing bad job? In general, I'm going to bring my understanding. And then to the quadrangle 3, then 9.5, you're doing good in both sides. You can call it adaptive effect. One of the gene originated, they're doing good for both sides, for everybody in the population. You call it adaptive. But for this one, almost 60% of them showing sexual antagonism. Each one is significant. So another somatic driver, tubulin P GAR4, basically gives similar result, but they also detect other different type of somatic function. But the result distribution is the same. Most of them is sexual antagonism favor male and disfavor female in nature. And I want to show other way around. Other way is that we use somatic driver, NOS, to silence. In this one, we click AD gene, which have detected effect there, AD gene there. Other 6 genes have one sex significant, other sex is close to 0.0001. I don't feel comfortable. I say that might not be significant. And there, what you see in the other picture. The picture is the first. Most of the gene there in the germline cell, 55%, which count 44 gene, which is doing good in both male and female. And the second one that is doing bad in both male and female, that take about 18.8%. Good thing that they identify a part of a gene in quadrant 2 that is take about 50%. And they are showing the female advantageous and male detrimental sexual conflict. The first one basically have very few. So this is the other picture from somatic cell. Now it may tell you that our somatic cell, well, Drosophila somatic cell and germline cell, they are showing opposite different function, different role there. They argue against with each other. So this is overall all 3 driver, all 623 gene distribution. There you see that they actually have some misalignment, and in the green line, that's somatic cell, red line, that's actin, the tubulin, and blue is actin 5C. That reminds us, what about in the same sex? Does different tissue show conflict? Within the same body of individual, are they fighting against each other in nature in terms of selection coefficient force? Here, give you some example. We identify more than 50 such a gene, which show conflict between somatic cell and germline cell and between different somatic cell. Here is give you an example, like first one, gene 17440. Its age is 6 million years old. And in the female, the fetal effect in the driver 1, that is NOS, germline cell driver, which is minor 0.0748. That's 17% of a negative. And so use no, suggest that in nature, they are doing good for female, for the germline cell. For driver 2, the same gene have 47% of fetal effect, which is tested by tubulin. So then this one I normalize it, and that's the same conclusion there. You see that. So you see clearly, you see that. And 2 different sacs, when you're locking down their gene effect, they have striking. You even should not use the word significant. Use remarkable high conflict in different parts. And gene 2, 6 million a year, same thing, 49% of nose, and minor 0.25 of tubulin. So that is for male. So that's other way around. You read that the figure eventually last gene 31941, which is 6 million a year to 11 million a year, which is a Drosophila melanogaster subgroup specific gene. In the male, their driver 1 tubulin give it's about 27.7% of higher figures in the lab that suggest that before locking down, they're doing really bad job. But it is in actin 5C driver 2, that's another somatic driver function that is doing very good job, about 23.8% of minors. But I mean, nature doing a very good job there. So this first time remind us that in nature, not only conflict between male and the female and in the function, they also have a conflict different part of the body. In fact, when we're looking at the different sexual kind of different tissue, that immediately implicated that the tissue will not dancing in same pace, in same individual. So that is another thing that even at the individual biology level, we should not teach students that all our different body parts help the same goal. They actually also have different goal there. They also have different job there. Right now, I'll give you total estimation. So we counting only those genes have fidelity, absolute fidelity effect, positive or negative, higher or equal to 10%. That is very high, 10%. That means 10 generation, you can 10 or 20 generation get a fix. Very, very high. So we counting. So there we found that in the male, the line gene, which show somatic positive effect, germline negative effect. The sixth gene show that is germline positive effect and somatic negative effect there. That suggests that in nature, a male favor somatic cell. I top in that in favor male somatic cell. But in female, we identify 36 genes. Among them, almost all of them that showing positive germline effect in nature and showing negative effect in somatic cell. Only 2%, the only two show different way. So this picture of what we call tissue antagonism tell you that male, female have different interests. For male, the interest in the somatic cell, much more like a muscle, competing, behavior, something like that. For the female, they more care about their baby, about their children. Their function where they show you like that. So that is very interesting where we saw this work. So this morning, when I wake up, I suddenly comes a big picture to interpret all this data. So far, what do we say the difference between the set? Actually passed 3 stages. First stage is Charles Darwin in 1871 published this book, the second most important book that called Descent of Man and Selection with Relation to Sex. There he claimed in what he called higher organism, there are male-male computation, much more than female-female computation. Then if a female can survive from disaster in the baby, like infanticide, something like that, then she have all the choice to choose good male to add the mate. So that, of course, Darwin, understandably, he didn't know genetics, he cannot see. Later on, after that Bateman, in 1948, he did one simple experiment. We asked a simple question. Let's male-female to mate, to say how often their mating have respond of the number of offspring called the fitness. What he saw that when a male mating more, have more children linear into safflower. In female, often the mating some stage, no matter how many times they mate, they will not increase the number of children because he realized that the number of eggs is really finite. After you you consume all the egg, then mating doesn't work. So for that reason, sex means different between male and the female. So he was really happy in the paper. He said he find a secret why Darwin was correct. Why? But he provide evidence of fitness effect between that difference between male and female. Then sometime later in 1972, British colleague Parker in the Journal of Theoretical Biology, he proposed, he called anisogamy theory. By the theory, at the cellular level, he stated that even organisms evolve from a sex ancestor have kind of isogamy state, I mean male, female at the same side, then after the selection, only those sex which have bigger egg, which have a higher succession rate to become children to succeed at fitness higher. For those sex who only contribute a little, just DNA like a male, then they're going to evolve small sac and also multiple sac. So he pointed out it is this reason to determining the difference of sexual dimorphism at cellular level. In that way, he provided cellular interpretation for Charles Darwin and Bateman observed, but they didn't provide how genetically happened. So in this Apollo, Artemis, and Drosophila analysis, we're able to say a genetic basis there. Organisms actually do not have female choices. They actually favor male in somatic cell at cost of female. So that in that way, good gene hypothesis doesn't work. However, differently from Darwin said that they're not because there's no female-female competition. There is a female-female competition, but a different way. They compete for germline function to find a good cave, to find a better environment for their children to grow. So they have competition too. That just didn't realize certain gene, we test about that in germline locking down. And then the selection is so strong, organisms even like to cost their own part of organ, which is not matter for their direct growth. For example, for male, they can cause their germline function. Anyway, they already have so many sperm germline cells. Why do you care? So that for female, they like to cause their somatic function, but the egg is so precious. Therefore, they need to take more investment to the female. So in that way, suddenly realized that this kind of observation provides a genetic basis for those previous sexual selection in Bateman paradigm, in Parker's anisogamy theory that can interpret it more about the difference between male and female in Drosophila. All right, so that is, I want to talk with everybody. So I summarize it. Today we see that they're species-specific duplicated. They can quickly evolve essential gametal function. And duplication is likely mechanism to resolve or mitigate sexual conflict. Then using genomic screening, we show that sexual antagonism selection is a prevalent force to drive gene evolution. You can go further that those major diversity in the phenotype we see in morphology might not really come from adaptive. Might that come from this kind of genetic conflict between the sex from plant and the animal? I cannot talk about plant dialogue there. So, but however, I have to point out that it is also a kind of 20% or 10% of gene there to help organ to adapt to changing environment. That's enough because they work in different time scale. For sexual conflict, every generation has to care about that. For adaptive to evolution, then you have to work in the ecological time scale, tens of thousands of years, millions of years when the environment really change. That's what we say the difference between the different performance, different genes, because the time scale are different one. And then we showed strong evidence for the genomic level that there is a split sexual conflict between soma and the germ. we also show that strong evidence showing that there are tissue antagonism. So our life, not more than population, individual, even within individual, is not perfect structure. So that the world where we stay, there's a lot. This is about 3 postdoc, 1 graduate student, and leading about we do about 4 group in the 10-year completed data. In recent 2 years, we had the data. We also appreciate collaborator, my theoretical collaborator, Stefano Alisila and William and Kovo. There we work about Bayesian theoretical distribution proof there. Thank you very much.
Happy to take a quick question.
Hi, Lynn. That was a fascinating talk. I had a question about the early part of your talk where you mentioned that most of the new genes came from the male germline, which makes sense because there's more cell divisions in the male germlines. That would make sense. But the male chromosomes don't recombine. And you mentioned that unequal crossing over is one of the most common sources of gene duplications. But if the male germline doesn't recombine and that's the source of most of the duplication, what is the predominant mechanism?
That good picture there. I have about 3 lines of response. That's a very good question, man. So male have no recombination, how they can get that one.
Yeah.
And in this gene group, first of all, we have 20% of gene. They come from develop. They do not need that kind of.
So it's a different mechanism.
Yeah, different. Come on, i.e. they are crossover dependent process there. Secondly, so far our knowledge about recombination to supplemental gas, and we know that other species of sunflower perform differently.
Oh.
The gene we should have, most of the genes are older than their gas are single lineage. They are about 3 million years older. So for that reason, they might still come from ancestral gene recombination to data show up.
Oh, interesting.
So that's a very good question. Yeah, that's right.
Okay.
For the Apollo and Artemis case, do we have any idea what the ancestral molecular?
I don't know that.
For the Apollo and Artemis case, do you have any idea what the ancestral molecular function is?
Yes,
and,
we do.
and how it's partitioned?
No,
How that
yeah.
result?
They are actually related gene there. And when we did a work, we showed that they might involve in some meiosis stage in a cellular level. All the positive work are good. I understand patients who carefully study the action type C from the show that it is a gene which is decoupled that heterochromatic. With a gene, you can make heterochromatic open, i.e., it's genetic. They published that particular work in the Journal of Chemical Chemistry, GPC. Yeah, that's very good one. Next time I should be sure about that. Yeah, thank you.
I want to make sure we have time for the class to meet after this. So that's thank Dr. Long.
Oh.
Yeah.
I mean, when the fertility then grows, okay, see you.
Thank you. That was fascinating. I don't know what to get to there, but
Right on.
Other than.
Oh, I see.
There's a class.
So you're in class. And then.
As far as I know.
Yeah, so I can just wait for you here. Where?
But unfortunately, the room had some.
Okay.
Just over there.
So-
It's just